Thanks, Don. Pleasure to be here. One of the challenges of being a speaker in the order is you never quite know what your other speakers are going to do and whether they've given your entire talk. I'm very pleased to say that we must be in the same mindset, because I'm going to try to give you, in essence, some of the concepts that Bruce has given you. I'm going to give them in a slightly different way. I'm going to use a few more pictures, but we very much are on the same message. So I want to start off with these two pictures. We're talking about collecting bloods in the first instance because there's a genetic opportunity there, but again, picking up on the latter points of Bruce's talk, there are many other omic opportunities. The other person there is Victor McCusick, who pioneered the online Mendelian inheritance in man. So in a sense, the last several decades, 60 years for DNA and 30 years for omics, have given us the opportunity of doing this, finding out more than half of the genes for Mendelian disorders. And 45 and up is not the study to do that work. But because it's nearly all done, that's why the time is now right for 45 and up. So in the red are the number, so there are around 4,500 medically described Mendelian disorders. In fact, as we do more genetic and genomic work, we're realising that there are more, and obviously we almost invariably never see the embryonic lethal ones because they don't end up being seen. Around 3,000 of the genes identified. So over the past 20, 30 years, there's been a progression going from Huntington's disease, cystic fibrosis, on to several hundred of these genes being identified each year by very conventional technology. In the recent past, and this slide really finishes too soon to see where the uptake goes because it really is a progression upwards, using whole genome, whole exome sequencing technologies, there's been a phenomenal increase in the number of single gene disorders being identified. Single gene disorders belong to rare and unique families. They're not population cohorts. But I guess I'm putting this here to place the population cohort in that right time frame. The next thing I'd like you to do would be to think a bit about the genetic space. And genetic space is on these diagrams. On the vertical axis is how risky is that genetic variant? Is it disease-causing or benign? Or does it confer some risk? So at the top, you can see I've in the bolded red circle, have 3 Mendelian genes, mutations in which cause early onset Alzheimer's disease. So Alzheimer's that presents typically in the 40s or 50s, not in the 80s that we would sort of think of being a more dementia time. On the bottom axis, we have the frequency of that variant in the community. These rare Mendelian diseases are just that. They're very rare. There are only a few dozen families in Australia with this inherited condition. So very rare, but very significant. Contrast that to normal genetic variation, which is very common, but has very little impact. In the middle, I've done it on the next slide, I've marked the ApoE variant. The ApoE variant was discovered very early on in the Alzheimer's field. It's quite common in the community. Around 15% of us in the room will have a copy of that ApoE variant, and it gives an increased risk for dementia. We'll go on and talk about the other risk factors down here in a moment, but let's just look at ApoE for one moment. So the normal person, if there is such a thing, and that's what studies like 45 and up go and show doesn't really exist, it has a 3-3 variant. That's the normal, most common. So this is percentage affected and age of onset of dementia. So in this very old data set, the average individual was having a 50% chance for dementia at around age 80 to 85. If you carry the E4 variant, 1 copy or 2 copies in the green and pinky purple line, your age of onset was brought forward by about 10 years. So that's the effect of a risk variant. Whether it's smoking or whether it's ApoE, there's the effect of a risk variant. It's a single risk variant. What about my entire genome? What about all of those other common variants that may confer some risk, but not of that same magnitude? So that's been called a polygenic risk score, where you actually sum up all of those other variants. So here, in this particular analysis, is looking at ApoE plus all of the other genetic variants, the totality, and the fourth quartile in the sort of hazard score doesn't look too dissimilar, and when you go and look at the age, about a 10-year bring forward in the age of onset. So that's using all the genetic information at a very population level. What about if you go and pull that out to a much more immediate and instantaneous effect? So in this particular slide, we're again looking at the incidence of Alzheimer's, age on the bottom, ApoE and polygenic risk. The ApoE for risk gene is sitting here in this red dotted line. The population risk is down here. And in this case, these are annualized incidence rates. So at any given point in time, what is the chance that in this year I will get Alzheimer's disease? And so again, if you look at the population average, at any point in time, if I'm an 80-year-old and I don't currently have Alzheimer's disease, what's the chance I'm going to get Alzheimer's disease? About 2%. However, if I took the highest risk group here, back at age 70, I already am at that 2% risk group. So if I was worried about my cognitive health, I could use that information. And with one of the other studies, the Maintaining the Brain, that's also accessing 45 up as study cohort participants, maybe there's a way where you can actually identify people at greatest risk and then intervene, whether it's cognitively through exercise, whether it's pharmaceutically. There are opportunities that extra dimension of the genetic information can provide. Let's keep moving along. The way in which those risk genes have been identified was by a technology called Genome-Wide Association. So you're looking at the association of any one genetic variant against a particular disease trait. Although the slide here of at that time, significant associations of any one genomic variant corrected for multiple testing for any particular trait. This slide is probably about 7 years old, because the one of today is just black with dots. Because to date, that approach, Genome-Wide Association, has identified over 50,000 unique variants, all multiple tests corrected. So we actually know a lot about risk. The problem with risk is risk usually comes in very small increments, and in genomics, even more so. So I'm going to try to illustrate that for you by thinking about human height. Human height is one of the most highly heritable conditions. There are extremes, as you can see, and those extremes are typically caused by Mendelian conditions. They're so extreme that we can see the phenotype easily. You can go and study dwarfism, gigantism, and understand some of the molecular pathologies that lead to those conditions. But probably almost everybody in this room isn't in that extreme. We're in the population mean of a hugely quantitative trait, where there's a regression to the mean. The biggest impact on height is environment, is diet. Who are the tallest people in the world? Some of the Scandinavians, the Dutch. There is very clear association with populations where massive dietary improvements have led to noticeable increases in height. So there's a complex traits, an interplay between your genetics and your environment. Using the GWAS approach, a couple of years back, there were 700 odd variants at 400 plus loci. They explained about 20, those individual variants that were significant in and of themselves, these were studies on 200,000 people, explained about 20% of the heritability. Using the polygenic risk score approach, took that up to about 60% of the heritability. So you're starting to get a pretty good snapshot as to what was causing height. But these were all through those very small risk factors, or contributing factors. I mean, if height, you think about, I don't know, heightism seems to be in, so therefore a risk factor seems to make you lower. This year, a really interesting study was published in the Genetics of Height. And that's why I've used this example, because I think it now comes and shows why we want to do these sorts of things in 45 and up. They used a technology which looked at known but rare genetic variation. So perhaps I need to go back a little bit. All of this GWAS work has typically been done on variants that are more prevalent than 5% of the population. In this particular study, they looked at variants that were only prevalent at 0.1 to 5% of the population, so the relatively rare variants. Of those variants, they identified some 700 additional variants that contributed to height. But these are quite rare, but the population-wide. But the really interesting thing was that there were variants that contributed either 2 centimetres loss or 2 centimetres gain in height in an individual. Now, of course, you've got 700 other variants here, so it's going to be quite relatively unlikely that any one of us are going to inherit, all of these and none of these, and therefore be restored. But the really interesting thing is this was that, and I'm not going to do any details, but each of these were variants in known genes. Each of them were a variant that altered the activity of a gene, be it in bone morphogenesis, be it in proteoglycans, things that are involved with bone development and plate growth and so forth. So what you could actually see here from a population level study was not only things that made quite a significant impact, but that actually also had, if you can take the abstraction from being height and a more general trait to think of this as disease, if you could go through and do this at a population level, then you would say, oh, these are people at high risk for disease, and we actually understand what's causing that disease. So this is now a very treatable thing. So I think the genomics of height, where in this study they looked at 700,000 individuals, are starting to show the power of applying these things to larger population studies. Quickly switch over to bipolar disorder, a major mood disorder characterised by periods of mania and depression. It is also very heritable, but there are no Mendelian forms for it, so it is a complex trait with risk genes and environmental impacts. The GWAS approach had been relatively challenging. Large sample sizes here, many thousands, and very few genes. As the sample sizes have got larger, more risk genes are being identified. We've done some whole genome sequencing work on parts of the families we've been studying, and in this particular figure, which we used as proof of principle, we could show that disruptive variants, so a bit like that height example, disruptive variants in genes that would make an individual gene not function had a correlative effect on the age of onset of bipolar disorder, so that the greater the number of disruptive gene variants you had, the earlier your onset of illness was. So that becomes rather important because in the very last section of Emily's talk of studies using 45 and up is this new New South Wales Health Genomics grant, which is led by Jan Fullerton at Neura, looking at the genetic contributors, clinical course and pharmacogenomics of bipolar disorder, because 45 up is, well, firstly the government call was to use 45 up, but this is the sort of resource that you want to do these studies in because of the linked records, because of the ability to actually understand both contributions to diagnosis, to progression, but in particular to responsiveness to medication treatment. Lithium, an Australian discovery, is the first-line treatment for bipolar disorder, but only about a third of people really respond well. A third really don't respond at all. So being able to give some better guidance as to whether people would respond from a pharmacogenomic perspective would be a hugely valuable outcome of this study. In this study, we're funded for the genome sequencing, but not the collection, they left that to us, of 1,200 people. So we could just go away and do our little thing, apply for permission, but that seems to be a very retrograde step. Why aren't we contributing that to the total collection? So the medical genomics panel has already done some collections, other studies have done some small collections. We see these are some ways where we can start chipping away at this larger goal towards actually getting that information. Finishing at the place where Bruce took us, at the UK Biobank, where they're starting with a very similar cohort in some respects, but a much larger one. They did recruitment blood samples, they did GWAS, and that's been hugely valuable for many studies around the world. Currently, industry partners are actually looking at whole genome sequencing of that cohort. And again, some of those things that we see in those height examples, I'm sure will come out of the UK Biobank example. So I guess my questions really conclude, can 45 and up afford not to do this? It was in the discussion right at the beginning. Cost was always a challenge, so I think most people are now saying, of course we can't. You must collect the sample first. You can then have a debate about which particular analysis technology you use. Yes, they do come at differing price points, but that's most of life. But you need to reserve the opportunity, and that's collecting the samples. I use the phrase 50,000 or 250,000. I know there are many reasons why 50,000 should capture the cohort, but I have a sense that it's not actually describing we want to collect this information on the whole cohort. And certainly I'd advocate, if you're doing further ways of refreshing the 45 and up, in other words, bringing in a new lot of 45s to 55s, I'd be just going in and saying that this is a part of the process, and the ethical oversight and review of the study, which is now well established, I think, is part of the confidence that the community can take that their samples would be looked after and maintained with integrity. And of course, again, picking up on those final points about the other omics, doing this, having those samples opens the door for many other opportunities. So, many years ago on a project that started just in the building next door, we were surveying groups of people. They were just about to go out and collect the samples, and the individual leading us rang me up and said, "Peter, should we do genetics?" And my answer was, in a very religious fashion, "Is the Pope Catholic? Should 45 up do genetics?" It's almost a non sequitur to be asking that question at this point in time, but at another level, it's fantastic that the opportunity is actually here, and I, like many of my colleagues, very much support that as a valuable adjunct to the incredible resource that 45 up has been and will become. Thank you.